All right, well, welcome everyone again. Today's Zoomtime Knowledge presentation. If this is the first time joining us, do know that this is a free public lecture series provided by the James Madison University Lifelong Learning Institute. I have a few people on the call or are meeting with us today. Important to know who they are. Sue G is our program assistant. My name's Rodney Wolfenbarger. I have the pleasure of serving as director of the Institute. We are going to be your digital assistants today. So if you need something in the meeting, you can't hear, you can't see, you're having problems connecting fully, just send us a message either in the chat window or through our email addresses, which we also monitor in the background during the presentation. We will try to get you connected. And again, today you can be as fully connected as you want to be. That's always the case. So you're free to turn on your video if you would like. Your microphones are intentionally muted. We will unmute your microphones after the presentation for the Q&A session. But for the first part, we're going to have your microphones muted so that Anne can go through her prepared presentation and slides. And I should mention and welcome our guest presenter today. Dr. Ann Henriksen is a faculty emerita at James Madison University, where she was a professor in the Integrated Science and Technology Department for 19 years. And the title of her presentation today is Telomeres. What are they and why does DNA need them? And Ann's going to answer those questions for us, as well as any questions that you might have following today's presentation. But Ann, I'm going to turn it over to you. I want to say welcome and thank you for volunteering your time and your knowledge with our learning community today.
Let me just get out of here for just a second. So do we see my video? We do, didn't we? Do we see me? Do we see me?
We have your video and your screen share.
Oh, okay. Well, let me unscreen share for a second so I can talk to everybody. I want to thank Rodney for what has been heroic help. We were together for an hour and a half on Tuesday, being sure this is all going to work. And he's an invaluable source of information. And I applaud his efforts to keep us together as a community and give us fun things to do during this very difficult and challenging time. So thank you, Rodney, and thank you, Sue, for being his right-hand person. I want to thank all of you for coming. This is a pleasure and an honor, and I want to welcome you to my office. This is my office where I do my work thing. So I'm at my office home computer, and which is now set up to be a little mini Zoom station. So thank you very much for joining me today in this talk. This is my first time ever doing this, so if I seem a little nervous, I kind of am. But I've always taught in person, and I've been able to see people's eyeballs. And it's a little different when I can't look out there and see if people are getting it or gauge your body language. So please do feel free to send comments to Rodney during the meeting if there's anything you think you need to say that I need to hear. With that further ado,
just getting started.
What's that?
Just cut her off.
Can you hear me?
Yes, we can now.
Could everyone hear all the previous things I said?
We did.
Great. Okay, so without further ado, I want to get started on the talk. And what I'm going to do now is I'm going to put my PowerPoint slides up. And we'll see. Is everybody seeing my PowerPoint slide? Wave or something?
Not yet, Anne.
Oh, I guess I need to share my screen, don't I? Okay,
right.
That's probably the first thing I should do. I should probably share my screen. So I'm going to share my screen now. Okay, now we're going to see if the PowerPoint will go up there. Now, does everybody see the PowerPoint?
You're good to go.
Okay, this is awesome. Okay, so this is very high-techy. So we'll just go through this, and if anything comes up, like I said, send Rodney a quick chat. So Telomeres, pronounced telomeres, because that's how Elizabeth Blackburn pronounces it. And if that's how she pronounces it, since this is her discovery, that's how I'm going to pronounce it. Telomeres are very interesting. On the left, you have a little picture here of, and I do have this fancy pointer, so isn't this cool? I have a fancy laser pointer here. This is an artificial rendition of a telomere, but over here on the right, these are real telomeres. Those are somebody's chromosomes. And on the ends, there are the telomeres that have been fluorescently labeled. And this is called fluorescence in situ hybridization. Those little shiny things are fluorescently labeled telomeres. And if you notice, they're all on the ends of the chromosomes. And these chromosomes are clearly ready to divide because there's a pair of them in the shape of an X. So we see those little shiny things are the ends of the chromosomes. Okay, now I want to start off by giving the usual caveat that we give when we give a talk like this talk does not constitute medical advice. I am not an MD, and even if I was, I wouldn't be giving medical advice on Zoom. So I just want to be sure I say that up front. I try to provide you with helpful information, but I'm not giving you advice as to what you need to do. Okay, so the first thing we have is this fun thing called a poll. And I'm sure y'all have done the poll thing when you signed up to get trained in Zoom. So Rodney has a poll he's going to make available for us. And here's the poll question. It's a yes or no question. And the poll question is, have you ever heard of telomeres? So I'm going to say yes because I have.
And just a moment here, we have almost everyone's voted, and I'm going to share in the poll and share the results in just a few seconds with you.
Sure. Y'all are pretty smart people. I've discovered in my teaching at LLI that no one believes me when I tell them some of the stories about what happens in my LLI classes and how smart all of the people there are. So I'm sure a lot of you have heard of this.
And are you seeing the poll results?
I am, and I see that 54% of the people's, we're up to 68% now. That's more than 2/3 of y'all have said you have heard of telomeres. So that's fantastic. Okay, so so the answer is a lot of you have. This is great. That means it's something you know a little bit about already. Okay, so so this is an aglet, and I never knew this word aglet until I started doing this talk, because we've always heard telomeres compared to the ends of shoelaces. So this is a shoelace aglet right here. This is an aglet, and telomeres basically function like shoelace aglets for your chromosomes. And so the important question is, why on earth do your chromosomes need aglets? And that's a really good question. I'll tell you the story of why we've decided that chromosomes need aglets. So there's always been this problem once we started to understand how DNA replicates itself. That is, the term DNA replicates itself is fancy for DNA copies itself. And we've always known as DNA copies itself that there's this problem at the ends. We called it the end replication problem. And the problem at the ends, which we will go into great detail about over the course of this talk, is that every time we reach the ends in the DNA replication process, the ends get shorter and shorter and shorter. And so the question has always been, how do your chromosomes protect yourself against shortening ends? And this has been referred to for decades as the end replication problem. And so scientists have always been curious as to how your chromosomes protect themselves as they duplicate themselves and lose little bits of their ends every time it happens. So the people responsible for solving this problem for all the rest of us are these three individuals: Elizabeth Blackburn, Carol Greider, and Jack Sostek. And I'll tell you, I've seen YouTube videos, and of these, especially the women, and they're hysterical. They're great people. They're great sense of humor. They did a lot of their discussion about telomeres over beer. So this was a fantastic discovery. And for this discovery of telomeres and telomerase, the enzyme associated with telomeres, they were awarded the 2009 Nobel Prize. And the Nobel Prize in 2019 was worth 9 million Swedish kroner. And that translates at the current exchange rates today to $915,000. So they each got today's equivalent of $305,000. So I'm going to play for you the video of the awarding ceremony when they were announced at the Karolinska Institute in Sweden. Oops.
The Nobel Assembly at Karolinska Institutet today decided to award the Nobel Prize in Physiology or Medicine 2009 jointly to Elizabeth Blackburn, Carol Grader, and Jack Szostak for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase.
So there you have, that's an announcement that usually made at our time at about 2 o'clock in the morning. So, okay, so to tell you about telomeres requires me to tell you about a lot of things first. And I don't assume at any of my talks that anybody already has prior knowledge about DNA or anything like that. So I always try and start with the part about DNA that is important for you to understand before I get to the sort of pièce de résistance of the lecture. So I'm going to start by telling you about a few other things first, which is important stuff. I mean, the structure of DNA is important to know, just to know, because we all know so much about Ancestry.com now, and we always wonder what exactly is DNA. So in going through the talk today, you'll learn a lot about the structure of DNA that's kind of aside and interesting from understanding the role of telomeres. So we'll talk about first the structure of a eukaryotic cell, and we are eukaryotes because our cell have nuclei, and any cell that has a nucleus is called eukaryotic. COVID-19 is a virus. COVID-19 does not have a nucleus. The RNA for COVID-19 just kind of floats around inside the virus. Our DNA, on the other hand, floats around inside the nucleus, inside our cells. So that distinguishes us from bacteria and viruses that don't have nuclei. We'll talk about the actual structure of DNA in detail. We'll learn how it's structured and how it looks when it's inside of your nuclei. We'll talk about the organization of DNA in the nucleus, how it's organized, not like everybody thinks and not like the pictures always show. Then we'll talk about DNA replication, how DNA copies itself so that cells can divide and one of your cells can become 2 and then 2 can become 4. We'll talk about that process because therein kind of lies the reason why we need to have telomeres. Then we'll talk about the lagging strand and replication problem and watch a few videos on demonstrating that so we understand that completely. Then we'll talk about the role of telomeres in DNA replication. What exactly are telomeres made of? And we'll talk about the enzyme telomerase and its role in telomere restoration. And finally, we'll talk about the pièce de résistance, why we're here, telomeres, telomerase, health, and longevity, what the research shows. Is there any real oomph to the idea that if you could make your telomeres longer, you would live longer? Okay, so let's start with a picture of the eukaryotic cell. And most of this stuff we don't need to know for this particular talk, but I'm going to point out 2 parts of this cell. The first one is, that's the nucleus right there. And cells are round, so we made the cell round. And that's the nucleus, that purple thing. And right inside of it is the DNA kind of floating around. Now, DNA does not look like people think DNA looks. In fact, DNA in your nucleus actually looks like a bunch of angel hair pasta with little tiny itty bitty meatballs all over it. And we'll see when we get to the structure of DNA in the nucleus why that's true. It doesn't look like a bunch of X's in your nucleus. That's a misnomer. But it's important to understand that it does live in that nucleus, and that it does have a shape that we're not accustomed to seeing. Okay, so this is called a GIF, and what I want to show you is this is a GIF, and the reason it's a GIF is because it actually can do stuff. This is the famous DNA double helix. This is that thing that Watson and Crick discovered, for which they won the Nobel Prize, and for which Rosalind Franklin did not win the Nobel Prize because she had already died of ovarian cancer, and you can't give Nobel Prizes posthumously. But her X-ray crystallography and Watson and Crick's putting her crystallography together with all their calculations determined this three-dimensional structure of DNA. And notice that it has grooves. It looks like a ladder that's been twisted. It basically looks like a step ladder that's been twisted. And we call it a right-hand twist because it's a right-hand screw. Okay, now your DNA exists in you in 23 chromosomes. Actually, it's 23 pairs of chromosomes. You get one from mom and one from dad. But what I want to show you is an illustration of the chromosomes. This is a chromosome. That's a chromosome. That's chromosome number 1. And interestingly, chromosomes have GPS coordinates. There's the P arm and the Q arm. They have locations. All the locations have long number locations, just like GPS coordinates. So on any given chromosome, you can find a specific gene by its GPS coordinates. Maybe we should call those gene position coordinates instead of global positioning coordinates, gene position coordinates like GPS. So all of your chromosomes are very carefully mapped. So if I have a gene on chromosome 1, you have that same gene on your chromosome 1, about 99.99999% of the time. Occasionally genes kind of get lost, but most of the time, my chromosome genes and your chromosome genes and everyone's chromosome genes are on the same chromosome. That's how we know where they are. And down here we have the X chromosome, the big X chromosome, and with a little baby Y chromosome. Sorry, guys. So if you're a male, you get the Y, and if you get a Y and an X, and if you're female, you get 2 X's. Okay. And this is what's called these are actual chromosomes. The previous picture was a artist's rendition. These are actual chromosomes. They're stained. They're fluorescently stained, and you can see there's 22 pairs plus the sex pair, and it's just a male or female. This is a female because there's 2 X's right there, 2 the same size. If there was a X and Y, it would be a little bitty Y and a big X here, but because there's 2 X's, this is a female's chromosomes, but this is an actual fluorescence image of fluorescently labeled chromosomes. Okay. And just to give you an example of what a chromosome looks like, this is the X chromosome. And this is just part of the genes on the X chromosome. There's hundreds of genes on the X chromosome. But what I want you to pay attention to for the purposes of this talk is that at the top and at the bottom, there are genes. There are genes at the ends of your chromosomes. Okay? So right there on the ends, there are genes. And that's kind of an important point when we talk about telomeres. Okay, so what I want to show you now is I want to start by talking about the molecular structure of DNA. And this is the kind of thing that's fun to know, even if you're not talking about telomeres. In every class I teach pretty much on DNA, I start with a discussion of the structure of DNA. Okay. So the first thing that is the ladder sides. We call these, this is called the backbone. This is the backbone, and this is the other side of the backbone. And they're like the sides of a ladder. And the sides of the ladder are made up of 2 big parts, the phosphate group and the sugar. And they're alternating, phosphate, sugar, phosphate, sugar, phosphate, sugar. If you go down, you notice they're alternating. And the phosphate in sugar, this phosphate is also in RNA, but the reason why this sugar is special is because this is the deoxyribose sugar. This is the sugar that characterizes DNA. It's missing an oxygen. It's a 5-ring. See if you notice there's five members of this ring. This is a 5-ring sugar called deoxyribose. Okay? Now, the bases over here, the bases are the alphabet of DNA. This is your human alphabet right there. G, C, T, and A. That is the human alphabet. And it's also the alphabet for every other organism that lives on this planet that we know of today. Every organism that lives on this planet, their bases are, their alphabet is G, C, T, and A. And that stands for guanine, cytosine, thymine, and adenine. Okay? So your alphabet only has 4 letters. That's kind of interesting. Considering you have 3 billion base pairs, and your alphabet makes up 22,000 different genes, 22,000 genes make you, that it's only made up of 4 letters of an alphabet is kind of interesting. And the next thing is we call these pairs base pairs, and T and A always pair with each other, and G and C always pair with each other. You'll never find a T across from a C unless something's gone really wrong in one of your chromosomes. Matter of fact, that sort of thing leads to mutations, and that's a problem. So this pairing up, these base pairs are very important. Okay, so now it's also important to recognize that DNA is actually 2 separate strands. The ladder is connected in the middle, kind of like with rungs, but it's really 2 separate strands. And the strands are what's called anti-parallel. Now this becomes a very important fact when we talk about DNA copying itself. The fact that the strands go in opposite directions. That was one of the confusing points that led to the entire end replication problem, because DNA can only copy in one direction. So I want you to note up here at this top, I just circled it, there's an O up there. And I want you to notice there's an O at the end of the other strand, because on the right, the strand goes in that direction. But on the left, the direction of the strand is in the other direction, okay? Those strands are anti-parallel. Now, so you ask the next question is what holds the strands together? And that's a very important question because there are huge consequences to what forces would hold those strands together. It turns out that GC and TA base pair are held together in the middle by hydrogen bonds. Now, hydrogen bonds are not bonds between atoms. Okay, the bonds between atoms are much, much, much stronger than hydrogen bonds. If you've ever wondered why ice behaves the way ice behaves and water behaves the way water behaves, it's because the molecules of water and the molecules of ice are held together by hydrogen bonds. That's why water is such an interesting substance. And that's one of the reasons why water is to our universe what water is. It's a very special substance held together where the atoms, the molecules of water, the H2Os, are held together by hydrogen bonds, just like the molecules of DNA are held together by hydrogen bonds. Notice there are 3 hydrogen bonds holding the GC pair together and only 2 hydrogen bonds holding the AT pair together. If it wasn't for those hydrogen bonds, your DNA would be 2 loose strands floating around in space in your nucleus. Okay? Okay, so an important point about DNA is that it is made up of building blocks, and the building blocks of DNA are called nucleotides. And when DNA is copied, it's copied according to these building blocks. So I've circled the building block up there. The building block is 1 phosphate, 1 deoxyribose sugar, and 1 base. So in this particular case, there's the phosphate, there's the deoxyribose sugar, and in this particular case, for this nucleotide, its base is G. But these are all, you can see these are all, there's another nucleotide, then there's the next nucleotide, and the nucleotides are the building blocks of DNA. So that when your body copies DNA so that your cells can divide and make 2 cells out of 1 cell, the way it puts things together is it just puts nucleotides together. Okay, so that's the building block of DNA. Okay, so when you see DNA, and I don't know how this happened and why this ever happened, but everybody's vision, this is everybody's vision of DNA, to this X like this. I don't understand how come this misnomer has persisted, but that's not really 1 chromosome. That's actually 2 chromosomes that have already copied themselves. And this is not how DNA is in your cells the majority of the time. This only looks like this right before DNA is ready to divide into 2 cells. Okay, this is how DNA really looks in your cells. So up there we have the double helix, and that double helix up there wraps around 8 proteins to make those little balls. Now, when I said that DNA in your nucleus looks like angel hair spaghetti with little meatballs all over it, that's what I was talking about. It looks like this over here, mostly like this, with these little meatballs all over it, these nucleosome meatballs. Okay? That's mostly what DNA looks like in your cells. However, sometimes it wraps up together. If your DNA doesn't need to be used in that particular cell, maybe this is an eyeball cell and you don't need your liver cells to be functional. Maybe the part of your DNA that controls liver cells doesn't need to be used in your eyeballs. So it might wrap itself up into chromatin. So it coils. So now we've coiled it twice. We've coiled it around the nucleosomes, and now we've coiled all the nucleosomes. And furthermore, if it's never going to be used, usually it will coil into the solenoid shape, 3 times coiled. So DNA coils up like this if it doesn't need to be used. And if it does need to be used, it looks like the angel hair pasta with the meatballs on it. Because we need to access your DNA. And we can't access your DNA to get stuff done if it's all coiled up. Okay? So I'm going to tell you some gee whiz fun facts about DNA now, which blew my mind the first time I ever heard them. And by the way, that thing I just circled at the top, that's a chromosome. Okay, so DNA, when it's ready to divide, looks like this. And that's how we know it's ready to divide. It's all super coiled. It's triply coiled, and now it's ready to split. Okay. Every cell in your nucleus has 6 feet of DNA coiled up so that it will all fit into that nucleus. So every single cell in your body, of which there are 37 trillion, has 6 coiled feet of DNA in it. That's a lot of feet of DNA. That's as tall as a 6-foot man. You have about 100 trillion cells in your body that contain DNA. And if we only have 37 trillion that are us, what are the other 67 trillion? And by the way, trillion is 14 zeros there. The other 67 trillion is the DNA of your microbiome. The viruses and fungus and bacteria that live in your gut and on your skin and in your mouth and in your ears and behind your knees and under your armpit. So we have about 100 trillion cells that all have DNA, 2/3 of which is not our own. If all the DNA in your body, including all your microbiome DNA, were untwisted and uncoiled and laid end to end, it would reach to the sun and back 600 times. And I did not believe this, so I did the calculation myself. And I came out with 610 times. So that's a long string of all the DNA in your body. DNA is part of the cell division process. You can't duplicate a cell unless the DNA duplicates, because you don't want to like take the existing DNA and split it up so that, you know, one cell gets 20 chromosomes and the other cell gets 3 chromosomes. You have to faithfully duplicate the DNA so that each cell gets its fair share of DNA, which is an exact duplicate of the mother cell that created these 2 cells. And cells divide in your body continuously. They're always dividing. Even when we grow up and we don't have to get taller anymore or get heavier anymore or grow new parts in the uterus, our cells are still continuously dividing and replacing themselves. So, but before a cell divides into 2 identical cells, the DNA has to replicate itself so that each daughter cell gets an exact copy of all 23 chromosomes. So here's some DNA. After DNA has replicated into a pair of X-shaped chromatids, and now it's replicated there, the cell is ready to divide. And each daughter cell will get 1 chromosome. And the DNA separates at this thing in the middle here called the centromere. This is called the centromere. It's where the DNA X is connected. But DNA separates from that. Right down that centromere, it separates. And the both halves go their own way into the daughter cells. Now we're going to watch a video about this in a minute. So we have some cool videos today. Okay. So here's a video about cell division.
Localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the 2 poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell.
Okay, so after this happens, then the DNA regroups, it forms a new nucleus and becomes its old self again, looking like spaghetti and meatballs. And the cell starts to furrow in, and eventually you end up with 2 separate cells, each having identical DNA as the mother cell. Okay? Okay. So now we're going to look at how DNA replicates itself. We're going to examine the process because this is where we start getting into why telomeres are important. So this is DNA, and DNA splits apart. It starts the duplication process by splitting. Okay? Because you can't duplicate each half unless you split apart. We'll have a video on this in a minute, but I just want to go over the basic ideas first. Then along comes this ginormous polymerase, and I drew that little thing, the DNA polymerase enzyme that brings in nucleotide building blocks. And it brings in a nucleotide building block, and it moves on to the next one. And it brings in another nucleotide building block, and it moves on to the next one. And it does that down the whole strand in pieces. And then after it finishes, and then simultaneously on the other strand, the same thing is happening. But what did we learn about the direction of DNA synthesis? It has to go in the other strand in the reverse direction, because we learned that you can only build DNA if from this end, the 3' end in this direction, and the 3' end in this direction. So that's where the end replication problem originated, because you cannot do this duplication process unless you first lay down a special molecule called a primer. DNA polymerase cannot just randomly start adding these nucleotides unless the primer is laid down first. So you have to have some DNA to make some DNA. And therein lies the heart of the end replication problem, which we'll learn about in a minute. So I just flipped this on its side, just so to make it a little easier. And now I'm going to make a ladder that kind of is a cartoon replication of DNA. Notice I put the 3' end here and the 3' end over here. And I put its opposing complementary strand in the reverse direction below it. And so that's a little graphical representation of DNA without all the complications. Okay. So now let's look at the DNA replication process. So first DNA splits apart because you can't duplicate it if it's attached. Then the nucleotides are going to get added, and there's my DNA polymerase zipping down the chain, adding the nucleotides. And I want to point out the primer here. The green stuff is the RNA primer that was laid down first because DNA polymerase requires this little starting bit before it can do its thing. So now we've done one strand. Now we have to do the other strand, right? Okay, in that direction, right? Oops, can we do that? We can't do that. You can't build DNA in the 5' to 3' direction. That's not possible. So the question becomes, how do we solve that problem? If we can build DNA on the 3' strand going in this direction, we have to go in the other direction on the other strand. It's kind of constraining, but Mother Nature has found a way around that problem. And we discovered how DNA duplicates itself before we solved the end replication problem. And that's why the end replication problem arose, because we knew these things and we didn't know how DNA did its job completely. So now we're going to watch a video. Oh, by the way, that bottom strand is called the lagging strand. So we're going to watch a video about the process of DNA replication.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment.
So that's all so clever that Mother Nature has figured out, is you make it in one direction smoothly and the other direction in bits and pieces. But don't forget that to make any piece of DNA duplicating, You have to lay down that primer first. You have to have DNA to make DNA. Okay, so some facts about DNA replication. DNA is forced by a special enzyme to separate at the two strands at many places on the chromosome simultaneously. Each strand is copied separately by an enzyme called DNA polymerase. DNA can only add nucleotides in the 3' to 5' direction on the template strand being the original strand of DNA that's being copied. And the enzyme requires a starting sequence called a primer that attaches to the DNA. But recall, DNA goes in opposite directions, so this presents a problem. DNA copied on the lagging strand will probably lose part of its end with every cell division. And why does that happen? That's the reason why it happens. Let's look at another video. That for a second. Where are you going to stick the primer? Out in space here? Like, are you going to put the primer like out in space? This part of the DNA is not going to be copied. Not now, not ever. Unless we find a solution to this problem. So could our chromosomes, now remember, we said there are genes on the end of your chromosomes. Could our chromosome actually lose parts of our genes that we need as we get older and our cells continually divide? Then this was the problem facing scientists. They said, God, do we lose our genes over time? What protects our genes from disintegrating away every time cells divide and that little itty bit on the end doesn't get duplicated? So that every daughter cell has a little less DNA end than the previous mother cell. No, thanks to telomeres. And telomeres was the discovery that solved the problem. Why don't we keep losing little bits of our chromosomes to eventually the point where our genes start to erode? I mean, imagine if that was a gene for your eye or a gene for your part of your brain that got eroded because the end of the chromosome couldn't be duplicated because of the end replication problem. So telomeres, what are telomeres? Okay, and that's kind of the important question. Telomeres are very, very long repeats of a specific base sequence. It's actually 6 bases. It's either 6 letters of A, T, G, and C that are at the ends of your chromosomes that do not contain any genes. And their main job is to be a buffer for the continuous loss of several hundred bases every single time that DNA replicates itself for mitosis. We have buffers. And those buffers are like shoelace aglets. So you can consider this to be a shoelace telomere. Or you can consider your telomeres to be chromosome aglets. So let's look at this again, the same video. We'll go over here to where we left off last time. Okay, so yeah, that was a cartoon of telomeres. Now I'm going to show you what they actually look like. Okay, so that's your regular DNA. That's before you get to the aglet. And I lined it up with the wrong numbers on top, but that's okay. So this is the long strand of one part of the telomere, and that's the other complementary strand. Notice I made one strand shorter than the other because that's exactly how it looks. And I've divided the telomere up into its repeats. And you can see the repeat is the repeat for mammalian cells is AGGGTT. And matter of fact, that is actually Elizabeth Blackburn's license plate. There's a picture of her license plate in one of the books that I've had. So telomeres are 6 base repeats. So it's this repeat again, this repeat again, this repeat again, this repeat again, and this repeat again, et cetera, et cetera. And I think there's up to 4,000 of those repeats on each end. And of course, there's the complement on the other side. For the A, there's the T, and for the G, there's the C, and for the G, there's the C, and for the G, there's the C, and for the T, there's the A. So the complement is on the other side, and that's what your telomeres look like. And there are no genes in your telomeres, okay? There's no genes down there. They're buffers. They're particularly there to protect against the loss, because as we'll find out later, telomerase doesn't work very well on you once you get born. There are only certain cells in your body that actually use telomerase. Pretty much the decay of your telomeres is something that happens over the course of your life. So the question is, does the decaying of our telomeres indicate anything about how old we are? Since telomerase doesn't work real well in most of the cells in our body, it only works in stem cells, in some white blood cells, and maybe one other kind of cell, but it doesn't work in all of our cells. So over the course of our age, our telomeres get shorter and shorter and shorter and shorter. So do your telomere lengths tell you anything or tell anybody anything about your age? In fact, they actually do. You can estimate somebody's age within a couple of years by grabbing a few of their cells, fluorescently staining their telomeres, and measuring how long they are. The older we get, the shorter our telomeres get. It is one of the primary physiological manifestations of age. It's kind of like tree rings, but in reverse. You can count tree rings for how old a tree is, and you can measure telomeres for how old people are. Some people have longer telomeres at the same chronological age. So telomeres are an interesting commentary on whether or not somebody's biological age and their physiological age matches their chronological age. In fact, longer telomeres are associated with a healthier, chronologically aged person that's biologically younger. Okay, these are pictures of actual telomeres, as we said from the first slide. We fluorescently stain the ends, and that's a complicated process that I won't discuss, but it is called fluorescence in situ hybridization. The abbreviation is FISH. But we stain them, and there they are. So like rings on the tree, the length of your telomeres can be roughly correlated to your age. So how many times do you think a chromosome can divide and lose part of its telomeres before enough is enough? Like, when do your telomeres get used up and then your cells go, oh my God, I can't divide ever again? There's an actual name for that. It's called the Hayflick limit. The Hayflick limit is a statement that says cells can divide about 40 to 60 times before they have reached the limit of their telomeres, and then the cells have to go into what's called cell senescence. It's like cell sleeping. Eventually, they undergo a process called apoptosis. And what apoptosis is, it's your body's way of self-orchestrating cell death. It's a scripted cell death process that your body will impose on cells that are old. It will cause them to rupture and eat up their parts and clean up the mess. And so that old cell that has too short of telomeres has been forced to die and is taken away. That's what apoptosis is. So there is a limit. Your cells can only divide a certain number of times before they've had enough. So now the important question for human beings, because if telomeres are an indication of how old you are, and long telomeres are good and short telomeres are not, can we repair our telomeres? Can we make our telomeres longer? Because if we could, would that be a fountain of youth? Well, of course, capitalism being what it is, has decided to set up a cottage industry to answer this question for us. And in fact, and medical science, of course, does research into this question. So let me first talk about telomerase, the enzyme that lengthens telomeres, which was that funny shaped blob on that video that kept making the end grow. Telomerase, because it ends in ASE, is an enzyme, is a complex of proteins and RNA, including an enzyme called telomerase. It acts to repair the telomeres. Now, some cells can repair their telomeres. Germ cells. Germ cells are our eggs and sperm. They're the things that give rise to our progeny. They can have their chromosomes and ends repaired. Embryonic stem cells can get repaired, but we've lost our embryonic stem cells a long time ago. And then certain white blood cells. We only have a few places in our body where there are any kinds of stem cells. But our regular cells in our body cannot, and under regular circumstances, do not repair their chromosome telomeres, hence they shorten with age. Now, it's very interesting to note that scientists have discovered that dysregulation of telomerase is associated with some disease states, in particular cancer. So telomerase dysregulation is not a good thing, even in under normal circumstances. And one of the manifestations of telomerase dysregulation being associated with cancer is that when we subject animals in the lab to telomerase supplements or to any kind of substance that increases telomerase, one of the serious side effects of that is that most of the time the animals do get cancer. Cancer rates increase statistically significantly. So you can't really play around with DNA like that without expecting some kind of cost. And usually the cost for playing around with DNA is cancer. So, but the question is not being rebuilt, but can telomere shortening be slowed? Can we live a lifestyle? Can we take a drug? Can we exercise so that our telomere shortening rates slow? And in fact, we can. We have discovered this interesting Eastern Chinese herbal thing called Astragalus membranaceus. And we have discovered that Astragalus membranaceus has the ability to slow telomere shortening by stimulating telomerase. And people in China have been using this herb for, millennia. But we have just now begun to understand that it has an actual, real physiological effect. Membranaceus, that thing that's often in Chinese herbs, they take it a lot over there for the immune system and various other remedy for various other conditions. But of course now the capitalists have latched onto this thing and made a cottage industry out of supplements that contain this particular substance. So the more important question to slowing down telomere shortening is, can telomeres be regenerated? I mean, it's one thing to slow them down. It's another question if we could regenerate them. So some scientists have taken astragalus, and they have extracted from the astragalus a subcompound called TA65, which they purify. And they've discovered that that TA65 is in fact the substance that stimulates the formation by DNA of more telomerase. So if you can purify TA65 from astragalus and you gave it to mice, would that affect their telomeres? Now, this is kind of like the experiments that have led to the cottage industry, which we'll go over in a minute. I have a whole lot of slides on the cottage industry of elongating your telomeres. But this, I actually read this paper last night. I have the paper right here. I read the thing in gory detail. And this is an amazing substance. Notice the title of this article: the telomerase activator, TA65, elongates short telomeres and increases health span of adult and old mice without increasing the cancer incidence. Kind of the holy grail. Make yourself younger without increasing the risk of getting cancer. Now, this is one of hundreds and hundreds of papers that have been written, but this was a particularly good one. So I'll tell you what they did. They took some mice, female mice, and they divided them into categories. They had the old mice, the mature mice, and 1-year-old mice are considered mature. I guess for humans, that would be maybe 50s, 60s. And they also got some mice that they considered old. And I think for mice, 2 years is old. And that would be for people maybe in their 70s through 90s. So they had old mice, immature mice, and older mice. And of course, if you're going to have mice that you're going to do experiments on, you have to have mice, they're otherwise identical, that are called controls. So they got some control old mice and some control mature mice. And what they did was they gave the mature mice and the old mice of both control and treated groups fruit mash. Mice love fruit mash. I don't know what's in their fruit mash, but I know they like it. And in the treatment mice, they put a supplement, TA65. But in the untreated mice, the control mice, they didn't put the supplement. So we have 4 groups now. We have mature mice that are treated and mature mice that aren't, and old mice that are treated with TA65 and old mice that aren't. And we say in science, ceteris paribus, which means all other things being equal. So you have to have 2 groups, and the only thing you change between the treatment group and the control group is whatever you're doing to the treatment group. Everything else has to be identical. How long you leave the lights on, how many mice are in a cage, what you feed them, how old they were to start with. That all has to be identical except for the one treatment that you're giving. Otherwise, you're going to confuse things. So the first result, they did this for 4 months. For 4 months, they fed these mice these 2 diets. And then they stopped feeding them those diets. They fed them regular old food. Maybe they got their fruit mash, but nobody got any treatment in their fruit mash. First result. In the treatment group, the treatment increased by tenfold. By tenfold. Now, in biology, that's huge. In the liver of the treated group, to make telomerase, the enzyme that rebuilds telomeres, in the 2-year-old mice. And 3 months after they stopped feeding them, they measured this. And 3 months after they stopped feeding them the treated mash, the concentration of telomerase in the liver of the treated mice was tenfold greater than in the untreated mice. That's a huge increase for an enzyme. Now, they didn't find statistically significant increases in any other part of the body in this particular experiment, but they did find it in the liver. Another result, TA65 significantly decreased the amount of very short telomeres in the treated mice, in the 1-year-olds and the olds. In other words, the proportion of cells that had short telomeres was significantly decreased in the treated group as opposed to the controls. This means This means that the treated group actually had to have their telomeres repaired. And TA65 significantly decreased the biomarkers of DNA damage in the treated mice. In other words, over time, your DNA gets damaged. The longer we're here, the more abuse our DNA has to be put up with. And TA65 significantly reduced the biomarkers of DNA damage in the treated mice compared to the controls. Now, DNA damage is one of those things that comes about because of oxidants. And I know everybody has heard the term antioxidant. And what antioxidants do is they take down the bad molecules that cause DNA damage. So oxidants cause DNA damage, and antioxidants take down those oxidants. So clearly, TA65 has a way of fixing DNA damage when the mice took it. Now, there's an interesting sidebar on this. They did find an increase in liver cancer in the treated mice, which makes sense because that's what the telomerase was, right? But it was not statistically significant. In other words, it was a little bit, but not enough to say it's statistically valid. That means you have to go do more experiments. And I haven't done any research to see if more experiments were done. But the fact that they had quite a few mice in each group. It wasn't like 2 or 3 mice. It was, dozens. So they had a good statistical sample to work from. And they had a little bit of a liver increase, but not enough to be statistically significant. So I'd say it was not 0, but it wasn't significant. But it still wasn't 0. Okay, so what happened to the mice? And this is interesting. So this is kind of a laundry list of things that happened to the mice. And I'm dividing what happened to the mice into 2 categories. What happened to the 1-year-olds, the matures, the 40 to 50-year-old mice, and what happened to the olders, the over 65ers. So in the mature 1-year-old mice, the TA65 supplementation had the following statistically significant, measurable effects. It improved glucose uptake. Blood glucose levels were significantly less. It lowered blood insulin, which makes sense because if your blood glucose is lower, your pancreas feels no need to put out more insulin. So of course, insulin levels will follow blood glucose, and it did in this case. So we would say that this significantly improved the 2 most important metabolic markers, glucose uptake and blood insulin. It showed less accumulated fat in the liver at time of death. Now, one of the fastest growing diseases in this country is called non-alcoholic fatty liver disease. Non-alcoholic fatty liver disease is a very serious condition that results in cirrhosis of the liver that has absolutely nothing to do with drinking alcohol. It's a result of accumulated fat in the liver that causes cirrhosis. It's a consequence of our diet and the way that we eat and how much we eat and how often we eat. These mice showed less accumulated liver fat at time of death, which means they would eventually show less incidence of non-alcoholic fatty liver disease. This is crazy. You know how when we get older, they say the fat between your skin goes thinner? It increased subcutaneous and epidermal fat layer. Now, there's graphs and charts and bar graphs all over this paper. So I actually got to see, the real data. It increased the wound healing capacity in the skin. It actually increased the speed with which wounds healed in the epidermis, and it increased epidermal cell turnover. And this is the craziest thing. They plucked a whole bunch of hair on the mice of both groups, and then they watched how fast the hair grew back. And on the mice that were treated with TA65, the hair grew back significantly better and significantly faster. In the old mice, the ones that considered the old mice, it had the following effects. It improved bone density. They actually took the little mice after they were dead and they measured their bone density. We all know how serious bone density problems, and particularly in females. They also showed less accumulated liver fat at the time of death. So there seems to be a sustained effect for improving the metabolic markers associated with the kind of things that lead to diabetes, high blood pressure, metabolic syndrome for mice that take this supplement. This is crazy. It increased red blood cells, hemoglobin, and platelets in the old mice. Those are things that actually go down as we age. However, one really interesting effect of this, and I saw this data and it was crazy, is it did not affect lifespan. All the mice died the same they would ordinarily die. But what's interesting was they might have died at the same time, but while they lived their life, it was a much better life. I guess from a point of view of health, at least. So it may not make us live longer, but it might make us live better. If we could make if we could increase our level of telomerase, that would cause our telomeres to be restored. Okay, so now this is where we get to the point where capitalism says we'll set up an industry to bring your telomeres back. Da-dum! Lengthening your telomeres brought to you by Amazon.com. I'm not making this up. Okay, you can do this when we're done here. I got all this stuff on Amazon. Okay, Elizabeth Blackburn's book. This is the book that she wrote about telomeres. Here we go. Books, more books. These are all on Amazon. I can't attest to any of them as far as their credibility, but there's a cookbook. Look at this, a cookbook. You can cook. Well, let's go to the next slide here. You can cook. You can cook for your telomeres. I don't know what that involves. I'm going to say Mediterranean diet, but maybe I'm wrong. You can buy supplements. Telomere support multivitamin. Telomeres up. Maybe they should say telomeres long or telomeres more. But this is all available on Amazon. Here's a supplement on Amazon. Telomere Health Dietary Capsules. And look what it has. Astragalus extract. 10% astragaloside. That's the thing that they fed the mice. That's kind of interesting. Here's another one. Healthy cell telomere length. Now, I noticed with this one, by the way, costs $35. This is Amazon's choice. I don't know how Amazon chose this. They did their own experiments or not. I don't know. But this has no astragalus in it. This has a bunch of herbs. This claims to increase the length of your telomeres, but there's no astragalus. I can't vouch for any of these ingredients. It does have milk thistle, which is actually very good for your liver, also named silymarin, which is right here. But I don't see any astragalus. But anyway, so this is another thing you can buy on Amazon. I put this slide here to show you that this has no astragalus in it. That's the previous ingredients. There's ginseng. That's supposed to be good for your brain. I have no idea what beetroot does. I don't know what any of those other things do. Oh, it has resveratrol. That's the stuff in red wine. This is great. That's the stuff in red wine. I don't know how much red wine you'd have to drink to be this much resveratrol, but I don't know. We could probably all do that. Okay, so I guess I'm at the end of my talk now, and what I'd like to do is I'd like to open my talk for any questions that anybody might have.
Hey, Ann, we do have a few questions already in the chat window. If you want to activate your chat window, you will be able to see those.
Let me see now. How can I do this? Oh.
And I can read them to you as well.
I want to go side by side here.
And after we answer those, I will allow people to unmute their microphone.
Sure. Stop video, spotlight video, unpin video. Let's see. Well, this is not particularly helpful in telling me how I can go. Okay, yeah. Well, let's go ahead and just ask some questions.
I will. Ann, let me prompt you with what's in the chat window since those have been queued.
Yeah, I have my chat window open now, Rodney.
Okay. So let's start with the one from 12:56 p.m.
I see. Is that from Herb? Is that from Herb Myers?
Yes.
Hi, Herb. Well, glad to have you here. There appears to be a telomere at both ends of the DNA strand. Why do we need them at both ends if only one end needs them? Well, that's an interesting question. Because at one end, the top needs it, and at the other end, the bottom needs it. Because don't forget, the strands are going in parallel, anti-parallel directions. So at one end of the DNA, it's the top strand that becomes shorter. At the other end of DNA, it would be the bottom strand that became shorter. So each strand will have one part become shorter, and it's the reverse end. So yes, both ends will lose part of their telomeres, but it's the reverse strand. It's the reverse. Does that make sense? Does that help?
And then let's go ahead and just work your way through the questions,
Sure, okay.
please.
Here's a question from Rodney. Did telomeres have any effect on the quality of life? Well, we found out that clearly they indicate not only the quality of your health, but changing them appears to be able to change the quality of your health. So the answer to that question is yes. This is a super question. Does chemotherapy, which kills cancer cells as well as fast-growing normal cells, have a long-term effect on people's telomere age? You know, Lynn, I don't know the answer to that question. But if I was a scientist who was involved in the scientific effects of chemotherapy, that is most definitely a question I would be asking myself. And I am actually going to write that down because I might look into that. Because that's a super good question. Because anything that causes your cells to rapidly be destroyed and reproduce will affect your telomeres. So that's a great question.
Hey, Anne, I do want to tell you, I will save the chat window and I'll send you all of these questions after so you don't have to record them manually.
Oh, thank you, Rodney. That's great. That's so good of you. Wow, you're so good at this. Okay. If I manage to lengthen my telomeres now, would my hair color be restored? That's a great question. I'm going to guess based on what I know about hair color that if it could restore telomeres in your melanin producing cells in your scalp, maybe. I don't know if anyone's ever done that experiment, but certainly you could Google telomeres, hair color, research, and see what you get. That's a great question. If you could affect the telomeres, that the melanin producing cells in your scalp, you might be able to do that. The question is, why was research conducted only on female mice? Because this is only one experiment. I'm sure that other people have done experiments on mice. These experiments have been done hundreds of times on different mice. This particular experiment, they were looking at biomarkers of female mice. And we know osteoporosis and things like that are female biomarkers. Also, if you're going to do experiments on mice, you cannot mix males and females together in the same group and come to the same conclusions. So if you did these experiments, you would have to have female mice in groups and male mice in groups and treat them separately and measure them separately. So the male mice would be excellent subjects for this, and I'm sure that someone has probably done that. That's a great question. We all know that females and males are not, females are not like males, only missing a body part. This is a good question. I can make this paper available to you if you would like me to. PowerPoints of this, a subset of my PowerPoints will become available after this talk. And in that, I will also include the journal reference or maybe even a PDF copy of this article. And then you could actually see how much they fed the mice. Okay. And they always did it in milligrams per kilogram of body weight. So you could actually do that calculation yourself. And I think the average female is about 54 kilograms. So you could actually calculate how much of that you needed to take in milligrams. And that's a great question. How do dysfunctional telomeres create tumors? That's a great question. It's not dysfunctional telomeres that create cancer. It's the fact that the telomerase enzyme has the power to alter the gene sequence. And anytime your body starts messing with the letters A, G, T, and C, no matter how it messes with them, you run the risk of upsetting oncogenes and tumor suppressor genes. So what happens is sometimes telomerase may inadvertently alter a tumor suppressor gene, or an oncogene and stimulate cancer growth or stimulate the lack of production of a tumor suppressor. You mostly worry about telomerase upsetting the gene structure of other genes in the chromosomes. Also, anytime you are doing things to the DNA sequence, no matter where it's happening, you run the risk of messing with the production of anything. Now, there's a specific pathway that telomerase affects, and for those of them, if you've had a biology degree, it's called the MAP kinase cascade pathway. And the MAP kinase cascade pathway is the pathway that utilizes some of the things that cause cancer, in particular RAS cancer. And anytime you mess with telomerase that messes with the MAP kinase cascade pathway, which has a lot of tumor suppressors in it and oncogenes in it, you risk messing with the guts of growth and growth suppression. So it's both direct and indirect. You indirectly mess with the pathway that leads to the production of cancer cells. Negative side effects of taking it, I don't know. That's a good question. I've never taken it. I don't know if these supplements have any side effects. That's worth investigating. It's worth Googling. Except for cancer, I don't know if it causes an upset stomach or, pimples or weight gain or weight loss. I don't know any of that. But if it affects glucose uptake and insulin production, I'm willing to bet it affects weight. Oh, excellent question. Do you think metformin impacts the speed of telomere decay? Yes, I do. And why do I say that? Great question from my friend Shannon Connolly. Hi, Shannon. So glad to see you here. Yes, it does, because did you know that metformin has life and longevity effects? Metformin is associated with longevity and increasing longevity. We don't know why, we don't understand why, but it is, and it does, and I'm willing to bet you that has something to do with telomeres and telomerase. That's a great question to Google, metformin and telomeres or metformin and telomerase. From Bradley, we have the question, do I have any other products to show? These are all unregulated, by the way. None of these are regulated. And that's because supplements are not considered drugs. And so the FDA does not regulate them. They have their opinions. They'll give you their opinions. I'm sure you can find their opinions, but they cannot regulate supplements, particularly because they're naturally occurring. So you can go on Google, Amazon, and find any of these. You can do searches for best telomerase supplement, best telomere restoring supplement. I'm sure you'll find them. I don't have any products myself that I know about at all. Is the study of let's see, we have this from Kathy. Thank you, Kathy, for your comment. I appreciate that a lot. From Frank Jumper. Hey, Frank, good to see you here. Frank is actually in Charlottesville, so he's visiting from afar. The study of telomeres differs in Europe versus US. I don't know if it differs, but I know that Europe they're very different in their approach to how they do science, what things they study. I know a lot of their regulations may differ, but I'm sure that scientists all over the world are studying this with the same exact level of enthusiasm and hopes for making a lot of money. That's an interesting question. Would T65 benefit people suffering from diseases now being treated with stem cells? That's an interesting question. I can't see where telomerase, and I am actually a neuroscientist by training, so I do know a good bit about things like Parkinson's. I can't see where telomerase would affect anything to do with the process of the kind of dopamine cell death that causes Parkinson's. I don't know for a fact, but my intuition tells me not directly impacting the disease itself. Now, whether it would impact the stem cells that you use to treat the disease, the answer to that is that's possible. I'm sure if you go on Google Scholar and Google the term Parkinson's disease, telomeres, stem cells, and see if those three terms unite into a journal article. And if they do, and you want to send it to me, I'll interpret it for you. Kathy Connery says, Google result, metformin shown to activate telomerase. Thank you, Kathy Connery, for doing that. Yes, I guess our intuition was correct on that. Metformin is clearly and unambiguously associated with lifespan in much the same way, by the way, as caloric restriction. I don't know if you're familiar with the idea of caloric restriction, but caloric restriction has the effect of increasing lifespan in dogs by 50%. Metformin also shows similar longevity effects. So the link between telomerase and metformin, which our intuition said is in fact actually, according to what Kathy says, you're true. Yes. So do we have any more questions? Did I miss any questions? Let's see, from the Moraleses, it doesn't increase life, but it improves life. That's a philosophical question. Anything that improved the quality of my life for as long as I was here, I would be happy with. The philosophical question about whether people are entitled to longer lives, that's a whole different question. I don't want to live to be 120 if the last 20 years are not productive and fun. If I can't play my ukulele and talk at LLI and visit my friends and drink wine, I don't really want to live. So if it increases longevity, but that longevity is not positive, there's really no point. The good part about what these data show that increases the quality of life, but not longevity. I think that's a positive thing. So, great. Well, I don't have any other things to add. If anybody else has anything they'd like to add, I am delighted to have done this talk. I want to thank the people at Sunnyside, because the people at Sunnyside were the people who originally pegged me to give this talk. And I gave this first talk at Sunnyside. And then decided to adapt this talk with more stuff for here. So thanks you to the person at Sunnyside who asked me to prepare this talk for the members of Sunnyside. So thank you to them, and thank you to all of you for listening. Thank you, Rodney, for helping me. I don't know what I'd do without you. You were just such a help and positive influence on LLI and on me personally being able to contribute during this difficult time. So thank you to you and everyone in the audience for coming. And it's been a complete pleasure. Thank you.
Thank you, Anne. And everyone still connected, do know that we will be sending out a recording of this video. If there's anything that you would like to play back and revisit, Anne will share her slides. I will include that when I announce the 2 Zoomtime Knowledge presentations that will be presented next week. You will get details by email. We will have one next Tuesday and one next Thursday. But as Anne said, thank you for attending. Hope you have a great day. Remain well. And it's good to see you all. We hope to see you back next week.